Okay, guys, we're going to get started. Hopefully the others are going to pop in. So do you remember what I asked you to do? Who would like to show off their drawing? And since there's 4 of you, I'm going to ask all 4 of you to do it. So who wants to go first?
Mr. Dawson, I'm trying to find a parking spot.
That's fine. We'll give you time.
Okay.
Okay, Kyra, thank you. Ikeria, Brandy, you want to go? Okay, sounds good, Ikeria. Hey, Brandy, are you there? Maybe she's not really there. Maybe she doesn't really exist. I've seen her in real life before. Maybe we're all in the Matrix. I was just reading something. Have any of y'all seen the movie A Beautiful Mind? Okay, I can't say this, then I'll give it away. It's really good, really good movie. But it's makes your brain hurt. But I can't reference it right now because then I'll give it away. I can't do that. I don't know. It's not the newest of movies, so it very well might be. Okay, I kind of want to know. I'll look it up real quick. No, it's not. Let's see. But I think it's on Hulu, possibly. So it's not on Netflix, it seems. Okay, guys, I'll wait for Landon to get there. Maybe Brandy will show up. So for those of you that just showed your drawing, hey, Brandy does exist. There she is. Do you have your drawing of the Homosporous life cycle?
Not with me. It's at my house.
Okay. When we do Fridays, I want to see at least one of them. Okay, we're going to have Friday. I want y'all to do the Heterosporous life cycle. And this is not for a grade or anything. This is mostly because I want you to sit down and just kind of make it fit, because we're going to make it different with our Heterosporous life cycle now. And I want you to understand where the differences are and where the similarities are. So for Kyra and Ikeria, did you find it helpful in any way to sit down and draw it yourself? In what ways? Okay, so Kyra says it was easier to make it go all together. Okay. Do you think you could draw it again from memory? Okay, that's one thing I want you to be able to do. And I need you to kind of analyze what's happening without having to look at a diagram. I want you, because if you can sit down and say, hey, I'm going to draw this for you, I'm not going to look at anything. I've got a sporophyte. The sporophyte makes spores through the process of spore mother cells. And then you work your way around. And then right now, being homosporous, we have one type of gametophyte, which makes egg and sperm. That's the key feature for us today. So, okay. Ikeria, do you have anything to add to that? Like how it was helpful to you? Y'all can use voice too. It's totally fine.
It helped me like see how one thing goes to another and just visualize all that going together.
Okay. Now, would you have done this had I not told you to? Probably not, right? So one of the ways that you can study for this, when we cover something, especially something like this, that we really put an emphasis on, going, how many of y'all have class like right after this class? I know you do. I think Elena does. She's got something at least. If you don't have time right after this, or maybe you have a class and then a break, as soon as you can, just sit down and work your way through these things. Okay, like today we're going to have the Homosporous life cycle. And um, if you will, what do you call it? What do you call it? I don't know. It's going to make it sit in your brain in a different way. Okay, now let me pull up the PowerPoint. I want to show y'all because I want to make a modification to what you've just done to show that as heterosporous. So, um, so we had the drawing in class. Okay, thank you, Elena. I'll see yours in just a second if you can. Okay, what we're going to do is modify only a portion of what you had to draw, not the whole thing. And that's something to keep in mind. A lot of times I'll say, like, here's homosporous, here's heterosporous, and you think of them as being two totally different things. They're not really. There's a lot of similarities in the way these two things live with minor differences, but those minor differences become very important. And that's where we want to kind of add those things in. Okay, so Elena, do you want to show off your drawing? Or I don't know if you're able to in the car. Okay, looks good. Looks good. So if you've got your drawing nearby, hold that out, look at it, okay? And I'll show you where we're going to make the modifications. So in this case, we're going to have two types of sporangia, not one. We'll have microsporangia and megasporangia, each of which are going to be on a megasporophyll. We've mentioned sporophylls, right? They're those little modified leaves where the spores are produced. So this is just going to be a megasporophyll, a little modified leaf where a megaspore is produced. The megaspore will go through megasporangia. So it'll have megasporangia. It'll develop, spores will develop from megaspore mother cells or megasporocytes through meiosis, making megaspores, megagametophytes. Will develop from those megaspores. Megagametophytes will have gametangia that only make eggs. So when you see mega in a lot of places here, are you going to think male or female? It's definitely going to be female. Okay. What do you think micro is going to be then? Going to be male. Okay. We'll sometimes have strobili that will be mixed. So if you've got some plants will have both micro and megasporophylls on the same plant. And we're going to see some examples of that coming up. So what I want to do is kind of go back to the whiteboard if I can get it switched over. Okay, so we started with a sporophyte. 2N, okay? From this, we produce either mega or micro sporophylls, so or sporangia. So let's do mega sporangia. They are also 2N at this time. They're going to go through, so let's say, let's do the micro. What do sporangia make? They make spores. And in this case, because it's a microsporangium, it's going to be microspores. What ploidy level are spores? Spores have gone through meiosis, so they are 1N. Do I need to write in meiosis here? Can y'all remember that? Let's just do it. Okay. What do microspores do? Microspores develop into gametophytes, specifically microgametophytes, which are, of course, through mitosis have become a larger organism that's still 1N. What do microgametophytes produce on them? They produce sperm, which are also 1N. Let's come back to the megasporangia. Megasporangia make mega spores, which are 1N. They go through meiosis. So let me type that up there so I don't forget. These will make, of course, megagametophytes. Which are 1N. Can't see. There we go. What do megagametophytes make? They make eggs, which are, of course, 1N. Your eggs, your sperm, come together through syngamy. Okay, and form a zygote. Which of course develops into your sporophyte. So what are the differences between the homosporous development that we saw yesterday and the heterosporous that we're seeing today? And it might be easier, instead of typing sometimes, because this is a long question, it might be easier just to click and talk if you can.
It doesn't have a spore mother.
Well, actually, it does. I just didn't put that part in. Okay, so that's a good question. Kind of like, okay, why did I leave that out? Mostly because I wasn't thinking about it. At what point would we put in the spore mother cells, megaspore mother cells, or microspore mother cells? Where would that go here if we were to include it? Sorry, guys, for putting in a little bit of an assumption there. Where would that go?
Right after the meiosis stuff.
So actually, it would be right before meiosis took place. So like in between the sporangia and the spores. In the sporangia, there are mother cells or sporocytes, because it's those specific cells that go through meiosis to make the spores. And I'll add that in right here. I'm going to do sporocytes just because it's a shorter word. Okay, but thank you, Elena, for pointing that out, because I could see how that could cause some confusion later. So that's not a difference, except that there's two kinds. So what else do you notice here? Okay, so Kyra said that there's two different sporangia going through different processes to make the zygote different from yesterday. Okay, so we got two different sporangia showing here, exactly. Okay, they are unisexual, if you want to think of it that way. So you've only got the female side or the male side. If you really just ignore that there's a male and a female side, those processes are no different than what we talked about yesterday, right? We have one split on yesterday's, and it's where you go from gametophyte to either egg or sperm, and then it comes back together again through syngamy, through the zygote. Here, the split is just back after the sporophyte. When you get into the sporangia, you've got two different kinds. So there's a lot of similarity, but what I'm trying to get at is, I hope it makes sense. Like, to me, it's very logical. If you're going to have a dedicated male side and a dedicated female portion, the process we talked about yesterday is exactly the same. It's just that we have dedicated parts of the plant, or sometimes separate plants, that are just for doing female reproductive portions, and others just for our male reproductive portions. They still are all part of the same life cycle. So we have these two kind of concurrent pathways, which can most of the time be on the same plant and just in different locations. Now we're going to look at some plants that do this. Most of our cryptogams are homosporous, okay, like we looked yesterday, but there are a few that are heterosporous that we're going through right now. So let me stop this share, come back to the other screen. Okay. Okay. So let's keep going. In these, hold on, sure I didn't skip something I want to talk about. Okay, we're going to talk about the different groups. I don't know why I kind of put some things in a little different order here. So we've got division Xylophyta. We've mentioned them, we haven't gone into them specifically. They are usually called the whisk ferns. They look like upside down brooms. Try a picture? Yeah. So like imagine like this is like a broom. They look like little brooms upside down you could sweep with. You know the old timey brooms? Maybe you've seen those. It kind of looks like that upside down. That's where the name comes from. And they have sporophytes, but they're mostly just little splitting, dichotomously forking aerial stems. They have no leaves, no roots. They have enations only, which are not really anything more than bumps. Okay. We have some in the greenhouse by accident. Some spores came in. I have had one sent to me and it died, and then I had some really nice ones, and I think they've died, but there's some other ones that have kind of spread around the greenhouse. So you can actually see these in person, okay? There are only 2 genera. This is an old group, okay? So they probably were more prolific in the past. There are 17 species, for example, the Psilotum genus. They are homosperous, making sense because they're so old. They would be early developing. And think about this, having no leaves, no roots, and they're being homosperous, those should all be clues that they're the closest things to bryophytes, probably, that we're going to talk about. They're very similar to the Rhiniophyta. Remember how I told you some of those extinct ones were going to come back? The Rhiniophyta comes back here because it's really similar to it. The gametophyte is non-photosynthetic. And so there's a similarity. It seems to be the way that it looks like Rhiniophyta, and then we've got this non-photosynthetic gametophyte. Now, we don't necessarily know how Rhiniophyta did that because the gametophyte would have been too small for it to be in the fossil record. But adding all of that together, these are things that are not features that we're going to see further on in plants. Stems with enations, and the stele is protostelic. Now remember, we talked about this. Protostele. Does protostele have pith or not? So if we were to do a cross section of the stem, we would have vascular tissue in the middle, and it would be a solid mass of vascular tissue. So no pith. Protostele has no pith, which we've already talked about too, is also being a very primitive form of a stele. Okay, so everything shows us that this is a very early plant. They have sporangia that are basically borne on the lateral branches. Notice that these are not necessarily like on the tips of each one of those. So if you go back and look at some of the early, let's see if I can do that. So when we look at the examples that we've got here, you can see something that looks similar in the fossil record. So not quite the same thing. Rhiniophyta has some terminal sporangia. We've got lateral sporangia. So I think Cooksonia is maybe one of the Rhiniophyta, but you can see something very similar here to those extinct ones. So this is the closest thing that we have to some of these extinct cryptogams. Okay, very boring looking plants, but I don't know, this one's kind of cool. I will tell you that this plant has become a little invasive in certain places, and I saw one of the most enjoyable scientific talks I've ever seen that was talking about this plant. And what was so funny about it is I've never seen someone have so much fun in their talk. You think about a scientific talk, someone getting up and being really boring with a PowerPoint presentation. Well, this one started out with a homemade dinosaur movie, which was just hilarious. Like they had these little fake, dinosaur toys, and they were like, rah, rah, and they had it in black and white, and it made it look like it was really old. And they were using these plants as the kind of background. And the point is that these plants have been around for a really long time. I'm not sure that they, well, probably they would have been around with the dinosaurs. I don't know. I haven't checked the dates for what people think about these, but we could look that up. You could see when raining off it is. It was probably around. But that's what they did in their talk. It was hilarious to watch. It was so much fun. And the reason they were doing this is because the campus where the talk was being was coming from was being invaded from different places by this species. It comes in on palm trees. It likes to grow in on palms sometimes and gets transported. And definitely those spores will spread instead of in the greenhouse. Okay, so here's one of those diagrams of their life cycle. Quickly looking at this, Homosporous or Heterosporous? Anybody? It's Homosporous. How do we know that? Well, we don't see any micro or mega words, right? Now, if I leave off all the words, how would you know? Well, you only see one type of gametophyte drawn, making sperm or egg. So Homosporous. Okay. Now, next group, division Lycophyta. These are sometimes called the ground pines, spike mosses, and there's a group that's really tiny called the quillworts. A lot of times we'll call these the club mosses as a whole. There's only two living representatives of two major genera. There's Lycopodium and Selaginella. I have some living ones in the botany lab. I have a hard time getting some of the club mosses to continue to grow, but I have some Selaginella. Their sporophytes have microphyls, so the tiny little leaf-like structures, tiny leaves that we've talked about that are protostelic. This group, though, has true roots and true stems, but they have protostele, so they're still very simple, okay? So early, probably developing. The Lycopodiaceae is the family that we put these in for the club mosses. You may see these in the woods sometimes. I haven't come across them a whole lot myself, but they do exist out there. There's 15 genera, 375 species of the club mosses. They are homosporous, so they have that bisexual gametophyte that will make an egg or a sperm. They have kidney-shaped sporangia, so that's a helpful clue. Their sporangia are specifically kidney-shaped to kind of, if you're not sure what I'm talking about with kidney-shaped, we're talking like, I think kidney bean. Here we go. And they have these oily, flammable compounds in the spore walls, which actually are, people do burn them, okay, for that purpose. So Lycopodium sometimes is called like the ground pines because they look like little tiny pine trees over here. They even look like they have cones, don't they? And so here's the interesting thing. Think about this. When we get to the cones later, do you think that there's a connection between what looks like a cone here and an actual cone? The answer is going to be yes. There is a connection. So they look like little Christmas trees. They have very, little simple stems, a few times branched, and they have rhizomes, so underground stems. Okay, looking at this one, is it homosporous or heterosporous when you look at it? It's homosporous, okay? Now you can see the strobilus here. I like this diagram of the strobilus. because you can see the sporophyll, you can see the sporangium, and you can even see the sporocytes inside. If you take this view and turn it sideways, you can see what it looks like this way. The microsporophyll has the sporangium on that leaf-like structure. There's a tetrad of spores. We won't talk about that yet, but we will come back to it when we get to flowering plants. Now the Selaginolaceae, say that one 5 times fast, has one genus, one genera. Well, one genus, I guess is more technically correct. Selaginella, 750 species. These are heterosporous. So I do want you to know the difference that Lycopodium, or the Lycopodiaceae, is homosporous. Selaginolaceae, the Selaginella, are heterosporous. We want to know that, okay? If you didn't catch my hint, you're going to be asked that question at some point. Okay. I could even ask you to draw me a general diagram of life cycles between these two. What would that look like? It would be your homosporous diagram for the Lycopodiaceae and your heterosporous diagram for your Selaginolaceae. Okay. So I may have you put those side by side as a drawing sometime on a test, on a quiz, maybe. Even if I don't ask, that's a great way to study.
We'll have to draw out like the gametophytes and everything.
So what we just did, we didn't have to draw any actual physical plants, but having
Okay.
all of the words that show homosporous versus heterosporous diagram. So does that make sense? Like we just did with the words.
Okay. I didn't know if you wanted us to draw the Lycophyta or.
No, I don't need you to come back and I don't need you to draw all these things because I feel like that's a lot of stuff to have on your plate. The only thing I might have you do, I mean, I might have you label something like a strobolus to understand like how all the pieces there. But I don't want to have you remember how to draw this gametophyte. That's not the point. So good question. So the microgametophyte, when it develops from a microspore, consists of an antheridium and a prothallal cell, which is vegetative portion of the plant. The megagametophyte consists of an archegonium and several vegetative cells, and these become a food source for the young embryo sporophyte. So these megagametophytes and microgametophytes are not very large. Okay, they have a sexually mixed strobolus. We will see this in lab when we get to that group. We'll actually look at a longitudinal section of the strobolus, and you'll be able to see that some produce microspores and some produce megaspores. So these are the spike mosses, very abundant in the tropics. They branch more freely than the ground pines. The leaves have something called a ligule. I'll try to show you a picture of that in a second on the upper surface. And they produce two different kinds of spores, which is why we say heterospory. So when you're looking at this strobolus, you can see those that are making microspores and those that are going to make megaspores. They're pretty obviously different, aren't they? When you look at them, they're very different. We'll see that under the microscope as well. The ligule, here it is a tiny little bit projection. So like if you were to pull this leaf back and look at the inside, there's going to be a little tiny thing on the inside here. Okay, so where you had kidney-shaped sporangia in the Lycopodia, here you're going to have this little ligule as a feature to look for. Okay, so I think everything makes sense through here. This is definitely heterosporous. We see two kinds of stuff on the haploid side of things here. Okay, I like these diagrams. I'm not going to ask you to reproduce them, except for maybe something dealing with or maybe labeling the strobili, those that are homosporous versus heterosporous, and some of the parts like the sporophyll. But I do want you to kind of look at these to be able to understand, like, if you were to find this and try to follow it along in its life cycle, could you understand if it's homosporous or heterosporous? Now, the last group, not usually talked about because it's very rare. This is Isoioides, and we do have Isoioides in Mississippi. but they're tiny. Look at the size of the plant when compared to the penny. You'll see that? It's very small, very, very small plant. It looks kind of like a grass, but it is not. It's usually found in areas, they're called quillworts, but I usually use the term isoides. Most are found in partially submerged water, at least part of the year. They have micro fills, like you see with the Lycopodium. And they're very tightly spiraled on a little tiny, tiny stem. They have ligules also, like we see in the last group. And they have something called a corm, which has a vascular cambium. Now, this is a big deal because a vascular cambium produces what? Wood, or at least it has the potential to produce wood. Are the cryptogams woody plants? The answer is no. So what does that tell us? That the development of the potential for wood definitely has been found in these earlier plants. Does that make sense? That's a big deal because we know that wood develops later from plants that are like this. And we're going to see that when we get to the gymnosperms. But as these plants develop, we don't have wood here, but we have something developing that potentially can cause wood in maybe later developed plants. Okay, so big deal that this thing has a vascular cambium. Now, I don't think I told y'all this. No. Okay. Homosporous or heterosporous? Hetero, great job, guys. Okay, so y'all were starting to pay attention to that, and I'm glad. Okay. Trying to decide if I want to go on into the next group today. I kind of want to have a separation actually between what we just finished and that. I don't want to rush into it because I want your minds to be settled on this idea of homosporous versus heterospory, okay? So Friday, I want to see a diagram of heterospory, okay? That's what I want to see. And I would like to hear from you if to tell me kind of what things, as you developed your drawing of your homospory versus heterospory, what are some things that stuck out to you? And it's okay if you say the same things as each other. Maybe draw it out and then go back and see some of the heterosporous versus homosporous examples, diagrams that we showed you. Because I want to get this set in your mind so that when we get to the next group, we don't lose what we've been talking about here, okay? That's really easy to do. You might be like, okay, I got this today, and then tomorrow we'll move on to another group and you don't have it anymore. So that's what we want to make sure we get stuck in your brains, okay? So this is a technique that you can do when you're studying. It doesn't matter what class you're doing, is you go and you review what you went over. So that's what this drawing is going to be, okay? You may even want to write out to the side which groups in the cryptogams are homosporous and which are heterosporous, okay? And then of course, Lycopodiaceae versus Selaginella, they're in the same division, right? They're in the same division. So we know that we're going to have to distinguish different Lycophyta from other Lycophyta. Okay, so Lycopodium, Selaginella, and of course we've got the isoides that we can separate out too. Okay, now if I talk about Psilophyta, the whisk ferns, we're going to talk about them as a whole, aren't we? Because they are exactly the same. And then when we get into Equisetum and we get into the Pterophyta, we're going to see that for the most part, they're overall going to be the same. It's the division that takes place. This difference is going to be in the Lycophita for the most part. Some Pterophyta, as we'll get to, but I don't want to get into that until Friday. I got to check one more thing before I let you go, because I can't remember. How much we've covered so far. I'm looking. I want to think about a quiz coming up. So alternation. Yeah, I think it's time for a quiz. So I don't want to have it on Friday. When would y'all like to have your next quiz? Wednesday?
I agree.
Okay, 2 for Wednesdays. Wednesday, good. Okay, that's for 3.
Wednesday's fine.
Randy's good. Kiviana, how are you? That's fine. Okay, so we're going to shoot for Wednesday. We'll do it like we've been doing. We'll Zoom it. I'll post it. Maybe we'll get to see cute babies. Come by and see us. This is awesome. When I first saw earlier, Brandy, I actually thought you were holding a baby doll because, like, from the angle, everything, they weren't moving, and I was like, is that a real one or not? Obviously, yes. Now, I was trying to figure out why you would be holding a baby doll. I was like, doesn't make any sense. Okay, so we'll shoot for Wednesday, and the things that are covered will be alternation of generations, your tissues for ground and dermal, vascular, bryophytes 1 and 2, and
You typed it in the box.
well, it's just everything since our last test, which was algae. So 9, 10, 11, 12, 13, 14, 15.
How many questions?
It'll be 10 questions plus a bonus, maybe, just like normal. So it's a lot of stuff to potentially be asked questions on, right?
Yes.
Kyra, I have put up the most recent videos, except maybe for, no, I think I got Mondays in. Can you check and see if y'all can see the videos? One of you at least. It should be there. Obviously not today's. I don't have that one up yet. But I think I got everything in Unit 2. And if you're not sure, like Tyler, like Unit 2 covers everything. So the videos in Unit 2 will help you know. So I got 10, 11. Yeah. Okay, everything's there. Great. I'll get this one. It has to render the video first, and then I'll get it posted as soon as possible. So Friday, we can talk a little bit about, and even Monday to some degree, we can talk about what you think might be on the quiz. Like, what are some things you should focus on? So remember those tables I've told you to start comparing? Put those together, okay, to kind of help you get things organized. I won't ask you everything. I can't ask you everything I want to ask you. Trust me. If I would not want to torture you all this way, but for me, I would enjoy just asking you everything. Like, there's so much stuff, and I find it so cool that I want y'all to know it too. But that's a little ridiculous. Y'all can't remember everything. So what we want to do is, like, what are the most important things to remember? And those are the kinds of questions I'm going to ask. Bonus, on the other hand, can be anything we've covered. Just so you know. Okay, but think about what you think is most important and ask if that's the case next week, okay, or on Friday. Okay, we can do that Friday and Monday. But we've got one more PowerPoint, and I'll decide. I may not put all of the cryptograms on that quiz just because it's a lot of information for the quiz, but the test, it will be. Okay. Okay, I'm going to let y'all go. Y'all have a great day. Take care. and I'll get this video up as soon as possible. Bye-bye, guys.